So, doctors, our today's topic is dental caries, recent understanding of biofilm and methods of management. As you know, dental caries is commonly known as tooth decay. Dental caries is an infectious microbiological disease of the teeth that results in localized dissolution and destruction of calcified tissues. It is according to study event. But other scientists, Schaeffer, gave another definition of caries. That caries it is defined as an irreversible microbial disease of the calcified tissues of the teeth, characterized by demineralization of the inorganic portion and destruction of the organic substance of the tooth. And I would like to mention you about World Health Organization classification of caries. In this classification, this shape and depth of the caries lesion scored on a 4-point scale. D1, clinically detectable enamel lesions with intact or non-cavitated surfaces. D2 or DK2, clinically detectable cavities limited to enamel. D3, clinically detectable cavities in dentine. D4, lesions extending into the pulp. Let's talk about histopathology. Caries of enamel. In pits and fissures, it spreads in triangular pattern with base towards dental enamel junction and apex towards surface, like in this picture. Microscopically, they define 4 zones: translucent zone, dark zone, body of lesion, and surface zone. According to Starter-Wendt, caries of dentine includes next zones: normal dentine, deepest area in normal dentine, which has tubules with odontoblastic processes and no crystals are in the lumen. Subtransparent dentine zone of demineralization of intertubular dentine. Damage to odontoblast process is evident, but no bacteria is found. Next zone, transparent dentine zone of caries dentine that is softer than normal dentine. Further loss of minerals from intertubular dentine. Many large crystals are present in the tubule lumen. Stimulation of this region produces pain. No bacteria present. And turbid dentine zone of bacterial invasion, widening and distortion of dentinal tubules, which are filled with bacteria. Very little mineral present, and collagen in this zone is irreversibly denatured. And infected dentine. Outermost zone consists of this zone. Consists of decomposed dentine that is teeming with bacteria, great number of bacteria present. Collagen and minerals absent. Next chapter of this lecture: recent methods of biofilm identification. Dental caries results from an imbalance of the metabolic activity in the dental biofilm. Many acidogenic and aciduria bacteria are involved in caries, supporting the notion that caries etiology is probably complex and multifaceted. The microbial communities of teeth have traditionally been studied by standard cultural approaches. More recently, cloning and sequencing of the 16S RNA gene have been used to characterize the microbial composition of the oral biofilm. The first molecular microbiological study appeared in dentistry based on sequence analysis of the 16th RNA genes only 10 years ago. As you know, this approach to evaluation of caries is so new, only 10 years ago. Next generation high through output sequencing method has the potential to reveal the composition and functioning of the biofilm by means of metagenomic and metatranscriptomic analysis. Important in understanding the dynamic processes in caries is the metabolic activity of the biofilm. Metabolome analysis is a new tool that might enable us to access such activity. Almost all molecular microbial analysis targeted at the identification of microorganisms are based on the 16th ribosomal RNA gene, which can be analyzed after DNA extraction from the bacterial samples. Real-time quantitative polychain reaction is mostly used nowadays because it allows both specificity for and qualification of pathogens. Next generation sequences. The open-ended approach, for example, no selection for specific species, that has been most widely used for oral microbial communities in the 16th RNA gene clone library approach. In this approach, the 16th RNA genes present in a certain environment and are amplified by PCR using universally conserved primers. The 16th RNA genes are subsequently cloned into Escherichia coli. In caries research, NGS has been used to analyze the oral microbiota in Chinese children with dental caries. It was a very interesting cross-sectional study, which revealed almost 42,000 unique sequences from 60 children with and without caries, indicated that the genera of Streptococcus, Veillonella, Actinomycetes, Granulocytella, Leptotrichia, and Theomonas in plaque are significantly associated with dental caries. Metagenomic provides, however, a tool to access the genetic information of a whole microbial community and to gain insights about the function of its members. It consists of the study of bacterial communities directly via the total DNA contact, therefore obviating the need for traditional culturing techniques and polychain reaction. And next approach, metatranscriptomic approach. Metagenomic approach involves the direct sequencing of the bacterial community total DNA. The methods for direct sequencing of oral samples were first tested on a dental plaque sample from a healthy donor and a donor having caries, employing the Roche biosequencing and the Illumina Solexa technologies. Individuals who had never suffered from dental caries had an overrepresentation of genes encoding antimicrobial peptides and quorum-sensing genes, among others, whereas samples from individuals with cavities had a high frequency of genes involved in functions such as iron scavenging and oxidative and osmotic stress. No metatranscriptomic studies of oral microbial samples are available. Let's talk about specificity in caries. So far, most of the molecular studies of caries, whether genomic or metabolomic, have been performed on saliva samples or on powdered plaque samples. Saliva samples are merely proxy variables of caries and may, at best, represent a reflection of what happens in the microbial community during caries lesion development in vivo. Dental caries is a localized disease of the teeth, and the caries processes are further modulated by intraoral and local environmental factors. Therefore, if we want to understand the etiology of caries, we must adopt a microbial sampling strategy that takes into consideration grading and compartmentalization in the oral cavity, site-specific difference in salivary film velocity, glucose and hydrogen ion concentrations, pH, composition of the microbiota. But please, notion that up till now, only a few molecular studies have adopted a site-specific sampling strategy for studying caries. And next chapter, etiology of caries. In etiology of caries, there are three primary factors: host, microbial flora, and substrate. But many scientists tell about fourth factor, time. Caries requires a susceptible host, a carcinogenic flora, and a suitable substrate that must be present for a sufficient length of time. This diagram shows correlation and interrelationship between different factors. Outer layer, the secondary modifying factor, but internal layer, primary modifying factors. There are tooth anatomy, saliva, biofilm pH, use of fluoride, diet specifics, oral hygiene, immune system, and genetic factors. And only in relation with all of that factors, caries can develop. And if secondary modifying factors will be present, risk of caries is higher. What about caries microbiome? As proposed by Mersch, excessive and frequent intake carbohydrates exceeds the buffering capacity of the healthy microbiome and leads to dysbiosis of the biofilm with change of the bacterial composition. In caries, continuous acid production from the metabolism of dietary carbohydrates favors the emergence of a highly acidogenic and acidiuric microflora. Enriched for Streptococcus mutans, other acid-tolerant streptococci, and lactobacillus species. Conditions of flow pH favor the proliferation of acid-tolerating and acidogenic bacteria, including Streptococcus mutans, lactobacillus, and other bacteria with a similar phenotype, while tipping the balance toward demineralization. It is only the biofilm-covered tooth surfaces that have the potential to develop carious lesions. Now about tooth habitats. Plaque communities and habitats, they're so different. For mucus, Streptococcus mitis, Streptococcus sanguis, Streptococcus salivarius. For tongue, Streptococcus salivarius, mutans, and sanguis. On teeth, non-caries, Streptococcus sanguis. Caries animal, Streptococcus mutans. Dentin caries, Streptococcus mutans and Lactobacillus. But in case of root caries, actinomycetes. And about role of saliva. The saliva flow rate and composition are well recognized as important host factors that modify the caries process. Saliva's protective role is in ability to clear carcinogenic food substances from the mouth, dilute, neutralize, and buffer organic acid formed by biofilm microorganisms, reduce the demineralization rate, and enhance remineralization by providing calcium, phosphate, and fluoride in the fluid phase of the biofilm in close association with the tooth surface. Oral clearance is slower for upper teeth compared with lower, and for buccal surfaces compared with lingual. The slowest clearance rate is on the buccal surface of the upper anterior teeth, whereas the fastest is on the lingual surfaces of the lower anterior teeth. This can be explained by the proximity of those surfaces in relation of the submandibular and sublingual glands. Studies have suggested that the saliva of caries-free patients contains higher level of immunoglobulin A antibody against many Streptococcus mutans epitopes than the saliva of caries susceptible patients. The organic phase of saliva contains antimicrobial agents such as secretory immunoglobulin A, lysozyme, lactoferrin, and peroxidase. Lysozyme can lead to bacteriolysis by disrupting bacterial cell walls. Lactoferrin binds iron, interfering with bacterial growth by both iron-dependent and iron-independent mechanisms. Lactoperoxidase can oxidize bacterial sulfhydryl groups, thus inhibiting glucose metabolism. Peroxidase protects salivary glycoproteins from degradation by bacteria. Salivary immunoglobulin A can react with Streptococcus mutans. Consequently, it may have implications on the prevention of dental caries. The organic phase of saliva is also constituted by proteins that may regulate the demineralization and remineralization processes involved in caries development and arrest. The statherines, proline-rich proteins, PRPs, histidines, and cystatines. It is suggested that those salivary proteins can provide protection against demineralization. But we need to mention about a role of diet. Dental caries has long been associated with the frequency of fermentable carbohydrate intake. Complex carbohydrates, or starches, are considered less carcinogenic because starches are not readily soluble in oral fluids and have a low diffusion rate in biofilm. Simple sugar, sucrose, glucose, fructose, are more cariogenic, with sucrose being possible the most. Therefore, it has been implicated as an important determinant of dental caries disease. The synthesis of extracellular polysaccharides can favor the accumulation of Streptococcus mutans and other carcinogenic bacteria in dental biofilm. The current concepts of dental caries focus on the fermentation of carbohydrates by carcinogenic bacteria producing organic acids. But when fermentable carbohydrates are present, the main organic acids produced are lactic, formic, and acetic acids. These acids coincide with a pH drop in plaque, resulting in demineralization of the tooth and creating an environment which is advantageous for further growth of Streptococcus mutans. Please, doctors, nota bene. Please remember that pH of 5.5 is the threshold for animal demineralization. Carcinogenicity varies with frequency of ingestion, physical form, and chemical composition. And of course, sticky solid carbohydrates are more carcinogenic. Sticky solid carbohydrates. Now let's talk about diagnosis of caries. Diagnosis of caries includes two main parts: caries lesion detection and caries lesion activity assessment. The assessment of lesion activity together with lesion detection is essential to arrive at the disease diagnosis and the appropriate clinical treatment decision. In addition to caries lesion detection, lesion or disease activity assessment must also consider etiological factor evaluating, such as oral hygiene, count of carcinogenic microorganisms in plaque and saliva, use of fluoride, sugar intake, socioeconomic aspects, such as family income and parents' level of education. The primary objective of caries diagnosis is to identify those lesions that require restorative treatment, those that require non-surgical treatment, and those patients who are at high risk of developing caries lesion. Conventional methods of caries diagnosis. First of all, of course, it is visual inspection. Visual inspection is the most common diagnostic method. Teeth should be clean, dry, and examined under a light source. In visual examination, changes in tooth structure such as enamel dissolution, white spot lesions, discoloration, surface roughness, and presence of cavitation are assessed. When illuminated, the caries tissues scatter the light and make enamel look whiter and opaque. This is due to increased porosity caused by demineralization. Similarly, when dentine undergoes demineralization, a shadow is observed under the intact enamel. When caries progress, the surface breaks down and the cavitation is formed. As can you see, a proximal surface of this tooth has caries. And in the International Consensus Workshop on Caries Clinical Trials, held in Scotland in 2002, the importance of the early detection of caries was emphasized, and the idea of development of international caries detection and assessment system, abbreviation ICDAS, was proposed. And in 2005, the ICDAS criteria was revised. It is picture from ICDAS Foundation website. Let's talk more detail. According to this classification, 0 score, it means sound, 1 score, first visual change in animal. Second score, distinct visual change in animal. 3, localized animal breakdown without clinical visual signs of dental involvement. 4 scale, 4-8, underlying dark shadow from dentine. 5, distinct cavity with visible dentine. And 6, extensive distant cavity with visible dentine. There shown another pictures according classification ICD-10. Score 1 looks like here. Score 2. Score 3. Score 4. Score 5. And score 6. If you like, you can found this research and read it yourself. Next point, next way, tactile sensation. The explorer is used for tactile examination, but the use of an explorer is not preferred because sharp teeth of the dental probe can produce traumatic defects on the enamel surface. The cariesogenic bacteria may be transferred from one tooth surface to another. Probing may cause cavitation and fracture in the incipient lesions. Explorers have low sensitivity, resulting in undetected lesions. Of course, we must remember about radiographic examination. And first of all, it is between projection. This X-ray is the most appropriate radiographic technique for caries detection. The disadvantages of radiography are as follows. Proximal contacts are overlapped, like here. The lesion depth may appear to be increased due to angulation, and this may lead to false diagnosis. Occlusal lesions may not be detected because of the superposition of the buccal and lingual cusps. Active and arrested caries can be distinguished in the radiographs. And doctors, please mention about cervical burnout. What does it mean? Cervical burnout is an optical phenomenon where a wedge-shaped radiolucent area is seen between the bone and the cemento-enamel junction. This effect is due to tissue density and the low penetration of X-rays at the cervical region. Please remember about cervical burnout because you can make a false diagnosis like cervical caries. Caries detecting dyes. You know that there are two layers of decalcification in caries dentine. The first one is the soft and infected layer which doesn't have the capacity of remineralization. The second one is hard, intermediately decalcified, and has the ability of remineralization. Caries detection dyes doesn't stain bacteria, but stain the less mineralized organic matrix. And there are two types of caries detecting dyes: enamel caries dyes. That one includes Protion, Calcine, Ziglo ZL22, and Brilliant Blue, and dentine caries dyes. This kind of caries dyes includes 1% acid red 52 in propylene glycol base and iodine. But in my deep opinion, you must know about novel methods of caries detecting. First of all, it is digital imaging. Digital image is an image composed of a series of sensors and pixels distributed orderly. On this picture, they are shown digital imaging and intraoral view after opening of caries cavity. The same situation, small spot, but so big caries cavity in vial. The same in case C and the same in case D. Device for digital imaging called DEXIS can view. Fiber optic transillumination. The light transmission index of decayed and sound tooth are different. When we examine the caries tissues with fiber optic device, we observe dark shadows along the dentinal tubules, as it has lower light transmission index compared with the sound tooth structure. Next method, digital fiber optic transillumination imaging. This method is the combination of the FOTI and a digital camera in order to reduce the shortcoming of FOTI. This system uses 780 nanometer wavelength near infrared radiation instead of a white light source. According to studies, this method is non-invasive, doesn't use ionizing radiation, and it is more sensitive than X-rays in detecting early demineralization. There are X-ray methods like xeroradiography, subtraction radiography, but I would like to tell you more detail about Mini-D. Mini-D uses LED and fiber optic technologies to detect occlusal and proximal caries lesions. The presence of caries is identified by two signals, sound and light. Green light turns to red one. It is also effective in wet environment. It is very important, but the plug must be removed before the examination. Next method, fluorescence. It is quantity light-induced fluorescence method, and they are shown a standard view view of tooth and view of tooth under fluorescence. And as can you see, the presence of proximal caries. Method called diagnodent. It is a special system. It has a range of 0 to 99. The value 0 indicates the healthiest state of the tooth. It is an effective method in detecting initial lesions without cavitation. Recently, a device called DiagnoCam was proposed to the market. The technology uses a laser diode of wavelength 780 nanometer for transillumination of teeth. And another new technology is Diagnodent pen from Cover Dental Company. This device works with the same principle as Diagnodent, and it comes with two different sapphire fiber tips, a cylindrical tip and a conical tip. In a study comparing Diagnodent and Diagnodent pen in detecting occlusal caries, it was found that this new device gives comparable results with DiagnoCam. These three devices are similar in effectiveness. Next method, carbon dioxide laser. The reason of the application of carbon dioxide laser as a diagnostic tool is because the subsurface of the caries lesion has more organic compounds than the adjusted sound tissues. When the carbon dioxide laser is applied to an incipient lesion, the organic contents evaporate, leaving a black carbonized residue behind, whereas the inorganic substance of sound animal containing minimum amount of water is less affected. Of course, ultrasonic or ultrasound caries detector. Ultrasound can detect lesions easily because the travel time of ultrasonic pulses differ in sound and demineralize animal tissues. This method is considered promising in detecting early animal lesions because the white spot lesions confined to animal produced to no detectable or weak echoes, whereas deeper lesions produce substantially higher amplitudes. And traditional X-ray methods like cone beam computed tomography. We can use infrared thermography for caries diagnostics and infrared fluorescence. And a very important part of caries management is prevention. What preventive methods we can use to prevent caries? Of course, firstly, it is oral hygiene and diet. Daily plaque removal by brushing, flossing, and rinsing is one of the best ways to prevent dental caries and periodontal disease. Patients should limit sugar snacks between meals. Next method, fluoride application. Fluoride prevents dental caries by inhibiting the mineralization of the crystal structures inside the tooth, and enhancing remineralization. Methods of fluoride application include water fluoridation, fluoride toothpaste, fluoride mouth rinse, dietary fluoride supplements, and professionally applied fluoride compounds such as gel or varnishes. So doctors, water fluoridation, it means drink water fluoridation. Next method, Pitt and Fisher sealants. As you know, every tooth must be sealing during 1 month after eruption, especially molars and premolars. And it is very useful and very important way to reduce caries level. Xylitol, one of the sugar substitutes. Xylitol has a sweet flavor comparable to sugar, but it is not only non-carcinogenic but also anti-carcinogenic. And, as you know, some scientists tried to develop caries vaccine. Some vaccines against microbes in the form of proteins, recombinant or synthetic peptides, or protein carbohydrate conjugates, as well as those based on DNA, have been successful experimentally. But however, none of these vaccines have appeared on the market so far due to difficulty in inducing and maintaining high levels of antibodies in oral fluids. Research is still ongoing for clinical. A very useful way to prevent caries is using calcium and phosphate compounds. The amorphous calcium phosphate and casein phosphopeptide. They're shown products of GC company, it is a Japan company, Tooth Smooth and MI paste. It's very effective things, very effective medicines to prevent caries, especially in pregnant women. Probiotics. How can we influence into caries level? We can modulate biofilm ecology. Modulation of biofilm pH. We need to know that a low pH is the main driving factor in the development of caries lesions. Physiologic factors that can counterbalance the acidification of biofilm include clearance and buffering capacity of saliva, metabolism of salivary substrates such as urea and arginine, which generate alkali in the form of ammonia. We can use arginine in caries prevention to modulate biofilm ecology. There are prebiotics, probiotics, and oral care formulations. L-arginine was identified as the main component responsible for the pH-raising effect of saliva by early in vitro studies. The major sources of arginine, L-arginine, for oral bacteria are diet, fruits, vegetables, and animal sources. Arginine as salivary peptides, de novo arginine synthesis from citrulline via protein turnover by the human body, using oral care products, for example, toothpastes and mints, and arginine supplements, for example, L-arginine capsules and protein snakes. In particular, ammonia production from arginine metabolism of oral bacteria inhibits tooth demineralization by neutralizing glycolytic acid and by favoring the growth of a desirable microflora that is compatible with dental health. Probiotics and prebiotics. Probiotics are defined as viable microorganisms that confer health benefits when administered in sufficient doses. Prebiotics are defined as fermented food ingredients that can change the composition and/or activity of the resident microflora and confer benefits upon host well-being and health. Some potential oral prebiotics such as xylitol, xylose, and arabinose could suppress the growth of Streptococcus mutans, but they could also promote the growth of some lactobacillus strains. And then let's talk about caries management. The treatment goal in caries management should be to prevent new lesions from forming, to detect lesions sufficiently early in process that they can be treated and arrested by non-operative means. If these attempts have failed, restorations will be required to restore the integrity of the tooth surface. Doctors, I would like you remember abbreviation like CAMBRA. This abbreviation is caries management by risk assessment. Caries management by risk assessment is an evidence-based approach for preventing, reversing, and, when necessary, repairing early damage to teeth. On this scheme, they are shown the caries imbalance between disease indicators and protective factors, the risk factors. And in case of decreasing quantity of risk factors, there will be no caries, but if risk factors will be increased, we will observe caries progression. And we need to know that when we tell about caries, we need to mention about identification of risk factors, look more detail. Risk factors. Caries risk assessment risk factors. First one, dietary habits, quantity and frequency of sucrose intake. Caries, past caries experience. Fluoride, decrease fluoride exposures. Habits, increase smoking, alcohol consumption. General health, use of medications causing xerostomia. Age, children and older adults. And of course, lower economic status and lower educational attainment. And risk indicators. Existing signs of disease process. Visible cavitation in pits and fissuries or in proximal surfaces. Brown spots, active white spots, or cavitated lesions on free tooth surface. And restoration placed in past 3 years. And assessment of caries lesion activity. According to these signs, caries can be active or inactive. As can you see on this picture, they're shown active multiple caries. And according to this table, we can compare, we can evaluate different factors and define low, moderate, or high risk of caries. As factors, they are shown caries activity, demineralized area. For example, demineralized areas. No white spots. It means low risk, caries risk. Inactive white spot, moderate caries risk, and active white spots, high caries risk. Or, for example, sugar consumption. If person eats sugar only with meals, it means low caries risk. 1 to 2 times between meals, moderate caries risk, and more than 3 times between meals, high caries risk. And according to this table, we can define a group of risk according to caries for every people. And we can define our activities, what we need to do to prevent caries in different caries risk groups. For example, if person is in a low caries risk group, we can use only over-the-counter fluoridated toothpaste or fluoride varnish for excessive root exposures or sensitivity, or sealants optional. But if patient belongs to high caries risk group, we need to use antibacterials, chlorhexidine rinse, and chlorine-based rinse, plus calcium phosphate supplements, plus twice per day. Use fluoride in 1.1%. Or fluoride varnish. And you can find in literature this very useful table where they're shown different methods and indications about caries treatment and techniques. What can we do? The first step in caries management starts with detection of the caries lesion, whether it is active or arrested. If the examination reveals arrested caries, no treatment is required. Please mention about it. No treatment is required. If the lesion is active, the general approach to this kind of caries should be preventive treatment. What does it mean? It means reduce sugar consumption or reduce frequent consumption by confining sugar to mealtime. Use sugar substitutes, plaque control, brushing twice daily with effective fluoride toothpaste. Use dental floss, application of topical fluoride gels, solutions and varnishes. Stimulate saliva by use of sugar-free gums such as xylitol chewing gums. If the examination reveals active caries but initial or incipient, the dentist has to think of a way to arrest the lesion, mineralization. Then restoration is never required. Caries on exposed smooth surfaces. Operative intervention is not required prior to cavitation. Even cavitated lesions can be arrested. Lesions which have plaque traps or deep should be restored. The general idea is, doctor, please remember it, clinically no cavitations. It means caries not progressive, arrested, no treatment. Caries progressive, antimicrobial and fluoride. But cavitated, we need to do restoration and of course antimicrobial or fluoride. And indication for restorative treatment. The tooth is sensitive to hot, cold or sweet. Occlusal and proximal lesions extend into dentine. The pulp is endangered. Previous attempts to arrest the lesion have failed and the lesion is progressing. The patient's ability to provide effective home care is impaired. And drifting might occur due to loss of proximal contacts. And of course, aesthetic reasons. So colleagues, thank you very much for your kind attention.